Hello, everyone, and thank you for taking the time to join us today. I'm very pleased to introduce Dr. Mike Samuel, who is the Principal Research Scientist and Scientific Liaison here at RainDance. Dr. Samuel received his PhD from University College London and performed Damon Runyon Fellowship-sponsored research at UMass Medical School. There will be a video recording of this webinar available on our website after the live presentation is complete. Please watch for an email from RainDance with a link to the video in the next few days.
Thank you very much, Chris, and thanks everyone for taking the time today to join our webinar. First, I'm going to give an overview of the RainDrop Digital PCR System, an extremely powerful tool for quantifying nucleic acid targets of interest. I'm going to be focusing on counting RNA. First, I will show you how to directly count input message RNA using a one-step RT-PCR protocol. Next, I will be outlining data showing viral RNA counting with an example using HIV. Finally, I'm going to show how this unique single molecule format enables one-step counting of microRNA. And I'm going to try to go through this talk quickly enough so that there'll be plenty of time for questions at the end. The RainDrop system consists of two instruments, the source and the sense, each with a disposable microfluidic chip that enables a wide variety of nucleic acid counting applications, including rare mutation detection, precise copy number variation counting, and aneuploid detection. You can quantify viral load, DNA methylation quantification, RNA counting, which of course is the focus of today's webinar. We also have recently released a new application for the RainDrop system, which enables sample preparation for sequencing low-input FFPE DNA samples called Thunderbolts. And there are many other counting applications the system can provide. The platform provides exquisitely high sensitivity and precision, multiplex analysis of each sample. The system is very robust and open to using many different reagents. It uses a simple and flexible workflow with a single pipetting step and incorporates contamination-free design. First, I'll give a brief review of exactly what digital PCR is. Digital PCR is a very simple concept that's been around for a number of years, but only recently has it been reduced to practice. The basic concept is that one divides the sample containing target molecules of interest into countable volume elements that either contain 0 or just one target molecule. In other words, the sample becomes digitized for counting. This is illustrated here with the complete sample containing the target in a complex background being divided such that there are a lot of compartments that contain no molecules of interest, and the compartments that contain a target will contain just one. Once you've divided the sample, at that point you use endpoint PCR to turn these single molecules into a countable compartment. At that point, it's simply counting. The number of bright compartments equals the number of initial molecules in your sample. The number of bright droplets can be used for an absolute count, and you can also count the total number of dark droplets, which gives you the volume that is being analyzed. Thus, division and digital counting of each molecule with an endpoint PCR assay provides sensitive, absolute concentration determination. So how does the RainDrop see it. Well, the RainDrop system is an amazingly powerful and versatile tool, which utilizes a simple, clean workflow. All one needs to do is load the sample onto a disposable cartridge. You can see here there are 8 wells for 8 completely independent experiments on a single run. The cartridge is placed into the source instrument, and the instrument automatically divides the sample into many, many partitions and deposits all of those into a standard PCR tube strip. At that point, you cap the tubes, and for the rest of the workflow, it's a completely closed system which prevents contamination from sample to sample or from sample to the environment. After capping the tube, you place the strip into a standard PCR, a thermocycler, and after thermocycling, the strip then is moved into the second instrument, the sense, where the fluorescent signal from each droplet is captured on 2 channels and then displayed for analysis. The key to this process is taking the sample and forming droplets with precise volumes of 5 picoliters. For example, if you pipette 50 microliters into the well on the chip, that sample is divided into 10 million droplets. 50 microliters divided by 5 picoliters. And only by having millions of compartments can truly digital counting be done. The dynamic range that's required for multiplex digital PCR of both RNA and DNA is provided by the RainDrop system. It really is just simple division and counting. Here's an example of how this works with a rare mutation DNA detection assay. Sorry about that. A sample containing EGS mutant DNA. This particular mutant, L858R, is very commonly mutated in several cancers. This was spiked into a high amount of wild-type background containing DNA. Together with 2 assays, one assay is Vic-based probe that hybridizes and detects the wild-type EGF receptor, and a FAM-based probe which detects the mutant allele. The data from one well is shown here, and what you can see is there are 7.4 million PCR negative droplets. These are the empty droplets that enable the other droplets to contain single molecules. There are droplets that contain single molecules of the wild type. There are about 920,000 single molecules of wild type. And at the same time, we can count independently, there are 46 molecules containing, droplets containing the mutant molecule. So you can see both low copy number and high copy number at the same time in the same sample. By keeping the DNA, total input DNA constant and titrating the amount of spiked in mutant allele, this titration curve is shown, and you can see a very linear response across 6 logs of dynamic range. This dynamic range enables measurement, as I mentioned, of both low and high copy number at the same time. It allows for extremely low limits of detection. It eliminates the need to find a sweet spot where you don't need to dilute the sample and do any pre-measurement. You can just load and measure. By having the sufficient dynamic range for creating these single molecule formatted droplets, assay multiplexing is also enabled. As I mentioned, there are 2 detectors on the system, and it's very straightforward to imagine how duplex assays, as was just shown, are performed where for target number 1, you have a probe which is, for instance, FAM labeled, and for target number 3, you have a probe which is Vic labeled. So you have, in this scatter plot shown here, fluorescence intensity for the Vic and fluorescence intensity for the FAM probes. And you can see a cluster for target 1 and target number 3, the duplex assay. But because the format is single molecule and the PCR is done to endpoint, you can also see 2 different targets using a single color by titrating the amount of probe that you pipette into the well. So for target number 1, a FAM-based probe was used at 1 concentration, let's say 25 nanomolar, and for target number 2, a FAM-based probe was used at, let's say, 50 nanomolar. At endpoint, the PCR reaction results in bright droplets for both of these targets, but target number 1 has half the intensity at endpoint as target number 2. And you can easily see these different clusters on the scatter plot. For target number 4, you can add 2 probes, both a Vic probe and a FAM probe, to the same target. And at endpoint, a droplet containing one of these molecules will hydrolyze both probes, and you can identify that in this cluster. And similarly for other multiplexes, if you wanted to see target number 5, you also would add 2 probes. In this case, the same amount of Vic concentration probe for target number 5, but twice as much of the FAM probe. Now I want to show you how this works for RNA counting as well. First, I'm going to show you experiments counting message RNA using a 1-step RT digital PCR protocol. To count RNA using qPCR, you need to add one additional step. You need to convert the starting molecule, which is an RNA molecule, to cDNA. And this is done routinely using a RT primer, a reverse transcription primer, and the enzyme reverse transcriptase. The RNA molecule is converted to the first strand synthesis, cDNA, and then in subsequent rounds with standard primers, that first strand turns into a duplex and is amplified. For qPCR measurements of the cDNA, you have 2 choices. You can keep those 2 steps independent and generate cDNA and then take that material and put it into a standard qPCR assay. Or you can do a 1-step protocol where all of the components are added together. Clearly, the 2-step method requires more steps and has potential to introduce contamination, RNases, as well as there are opportunities for users to make errors. The 1-step method, although simpler, sometimes doesn't work. For instance, there's no demonstrations of miRNA, microRNA, using 1-step methods. And both steps can show differential efficiency for the reverse transcription step. This is an area of active research and is very interesting. These limitations are completely eliminated by using the 1-step RT digital PCR protocol and the RainDrop. So the first time the experiments were run using a 1-step master mix, 4 different 1-step RT qPCR kits were purchased and worked immediately right out of the box. For most of the experiments that I'm going to be showing you, I picked just one of those, which was superscript 3 kit, very commonly used. And what we found was the same broad dynamic range, 6 logs, the same precision, and the ability to do multiplexing with color and intensity that we saw with RNA, all of these features are completely enabled for quantifying RNA. First, I will show you some of the data demonstrating the wide dynamic range and small fold changes that can be detected using this system. What you can see at the top are scatter plots showing two assays. One assay detects an exogenous spike-in material called Xeno, which is commercially available from Life Technologies, where you know exactly how many RNA molecules you're pipetting into the well. And there's an associated TACMan assay that goes with that. For this demonstration, I picked a gene expression assay for GAPDH, a common housekeeping gene. And what you can see on the left panel is that you can clearly count and detect the xenoRNA when there is no total RNA added. When you add total human RNA, this is material that was, again, commercially purchased from BioChain. This particular scatter plot shows 10.4 nanograms of human RNA input. You clearly can see GAPDH is able to be assayed and counted and is extremely abundant in this sample. On the bottom left, experiments were run where the Xeno inputs were kept constant at about 1,000 molecules, and the total amount of human RNA was diluted down across the 6 logs of dynamic range as we expected to be able to count. And as you can see, we have very nice linearity that goes all the way from higher amounts of RNA input, down to the regions here in blue, which are typical for the amount of RNA you would purify from fine needle aspirates, and even down to the range where single cells RNA is being counted. If you zoom into this region here in the dynamic range, replicate experiments were done to illustrate how you can see very small fold changes. So in this particular example here, 6 or 6.6 nanograms of total human RNA were added, and down at this range it was 1 or 1.2 nanograms. So a 10% or a 20% change in the amount of total input RNA was clearly able to be detected, and the CVs were down below 2%. So clearly the system is very precise and enables very small fold changes to be quantified. Next, I will show you how multiplexing is also enabled for RNA, similar to how it was shown for DNA. For this example, a quick 4-plex assay was put together using the Xeno exogenous spike-in RNA as a control with a BIC assay. 2 different FAM assays were used, one for GAPDH and another for PPI as housekeeping genes. And finally, Pol R2A was assayed using both a BIC and a FAM probe, and you can see that cluster here. The total amount of RNA was titrated across this range, and you can see the amount of each of these species identified here. And what you can see is that the amount of GAPDH and PPI molecules are almost identical. These lines are just about on top of each other, and they have the same slope. Now remember, this is absolute counting. There is no relative quantification. So these samples, total human RNA samples from BioChain, have similar amounts of these 2 species. For Pol R2A, there's about a log fold less amount per nanogram of total RNA. And again, you can see a linear titration with the same slope. These were pre-designed TACMan assays right off the shelf with no optimization. This is an extremely easy system to use and to put together multiplex assays. Next, I'm going to show some data counting viral RNA, using HIV as an example. Just as a reminder, HIV is an RNA virus with RNA encapsulated in a protein coat. And in the life cycle of HIV, these particles enter human cells, the RNA is unmasked and integrates into the host DNA and exists there in a latent form until whatever time the virus escapes latency, becomes RNA, and goes back into its reproductive phase. So there are key questions of interest to researchers in both aspects of this life cycle, HIV as integrated DNA and HIV as RNA, both during infection and as it escapes latency. So first, I wanted to show some data from a customer just to reiterate the value of having sufficient dynamic range for counting both high and low levels of targets in the same sample. So this was customer data where they provided total DNA that had different amounts of HIV contained. To see latent HIV, you would like to be able to see a very few number of copies of the integrated HIV in the background of a lot of wild-type genomes. And that's exactly what the system demonstrated. So this is 8 samples run in a single run, where you can see there are 2 assays on the Vic axis. There's an assay for a housekeeping gene, RT-30, and on the FAM axis was an assay designed against the HIV GAG molecule. There were approximately 1 million genome equivalents counted by looking at the number of RT-30 molecules, and this is tabulated below. And in this titration series the customer provided, it went from several thousand GAG molecules down to below 10. This is graphically illustrated here. This was not optimized, and this was just a single run, and you can clearly see linear counting down to around 10 HIV molecules per million genomes. Now, what about directly counting HIV RNA? For the initial experiments, I first needed to get the relevant material and assays, and I purchased Zeptometrix natural control reagent. This is a viral-like material that is used as a control in the industry. This was lysed, and the RNA was purified using a QIAMP viral purification kit. The assay was chosen from the literature to count, again, the HIV GAG RNA. And this came from a paper from Nature, 1992, from Danny Dewitt. The primers and probes were synthesized from IVT, and the GAG assay is a FAM-based assay here. So on the far left is data that shows that both the assay and the reagents work. Again, I've used the exogenous spike-in RNA as a positive control, where I know exactly how much material, how many molecules of RNA are in the assay, or in the sample. And you can see the HIV GAG assay works very well. The next 2 panels show spiking in either no HIV or similar 1 microliter of the HIV eluate from the purification, but now in the background of total human RNA. And what you can see, both visually and in the table, is that there is no effect of having the total human RNA on counting either the exogenous spike-in or the GAG itself. So the HIV GAG assay counts RNA accurately, independent of what total RNA is input. The second set of experiments was to go through a titration series using that same material, and that's shown on the left. In addition, there's other commercial material that is commonly used in the industry called Acrometrics that's sold by LifeTech Technologies. And that was similarly purified using the QIAGEN RNA kit. And either 0 or several microliters of that eluate were pipetted into the well in the presence of total human RNA. And for this series of experiments, instead of using the exogenous spike-in as a control, GAPDH was used as an endogenous control. So you can see endogenous RNA and the HIV GAG RNA in the same experiment. Again, you can see very linear response. These measurements were done in replicates, and you can see in the table below that the CVs are remarkably precise, very low CVs. Finally, for these control experiments, I put together a Triplex assay where 3 different assays were added together to the well. The GAPDH for the endogenous control, the HIV GAG, which is the molecule we're counting, and in addition, the xenospike-in was used where both FAM and VIC hybridization probes were used. And again, you can see very clearly the controls and the GAG is counted quite accurately. Finally, this was tested with customer samples. The customer provided both the assay and the sample material. They also used an assay for HIV GAG. And for comparison, I'm showing the assay from the previous slide that was the RainDance assay pulled from the literature. And this is the assay the customer provided. Again, very clear that you can count GAG, and there's a very low, in fact, a 0 background here. So the samples went through a dilution series of the Acrometrics material and also real plasma samples the customer provided. And you can see that the Acrometrics dilution series is highly linear, and also several of these samples were done in replicates, and you can see they're outlined here in red. The negative control, the low input Acrometrics positive control, and the plasma samples themselves were highly reproducible with low CVs. Finally, I want to go on to show how the single molecule format is enabling new capabilities for researchers that have not been able to do these types of experiments before. Specifically, this is for measuring microRNA. And a reminder, microRNA are approximately 22 base pair non-coding small RNA species. There are approximately 2,500 mature human microRNAs in miR base. They have diverse roles in transcriptional regulation. Interestingly, they're very stable in peripheral body fluids, and in fact, they're found in exosomes as well. So they have great potential for being biomarkers to measure a variety of disease states. Now, microRNA can be analyzed using qPCR. In particular, there are reagents sold by Life Technologies, TACMan microRNA assays, which have a unique feature that turns the short RNA species into a long enough species to use TACMan reagents. There's an RT primer that has a hairpin stem loop structure, and this is what primes the original cDNA first-strand synthesis. However, the microRNA qPCR assay has not been shown to work in one-step RT-PCR, and also has not been shown to work in multiplex. These limitations are now removed when using RainDance RainDrop technology. So the initial experiments, given the efficiency of RT-PCR, were really just to test out to see if this would work. And you can see this on the left, where synthetic RNA for Xeno and synthetic RNA for miR16 were put into the well, either in the presence of the RT primer or in the absence of RT primer. And you can see for the FAM-based assay for miR16, we clearly can count this with extremely low background. Similar to the previous studies, this was repeated now in the presence of total human RNA, again, with the RT primer or without the RT primer. And again, you can see this is very, very quantifiable assay. There's a lot of miR16 in total human RNA in this particular sample from BioChain. But in the absence of the RT primer, you don't count anything. The table gives you the specific numbers. Again, you can see it may be of interest to note that the number of Xeno molecules is highly reproducible here. So there's a low background without the RT primers. It's very specific. And there are no false positives or negatives added to the assay in the presence of total human RNA. Several multiplexing assays were put together. On the upper left, 2 different microRNAs, miR21 using a Vic probe, miR16 using a FAM probe, and the Xeno spike-in control using a mixture of Vic and FAM probes. Pol 2RA was used in the middle scatter plot as an endogenous control for microRNA. And then on the right, you can see miR21 as a Vic assay with miR92A as a FAM assay. These are very commonly dysregulated in a variety of cancers. And then down below, there's also some keen interest in the field of HIV in host responses to the infection. And so what's illustrated here is the ability to count miR21 and HIV GAG in the same assay. So you're looking at both endogenous host response and the viral infection in the same assay. Finally, some titrations using a triplex assay are illustrated in this slide. On the left, you can see the triplex assay, miR21 with a Vic probe, GAPDH with a FAM probe, and miR16 now with a mixture of Vic and FAM probes. And to illustrate the specificity, if you look below in the middle panel, only the RT primer for miR16 was added to the well. And you can see the cluster for miR21 disappears and doesn't have any effect on the other molecules you're trying to count. Similarly, if you remove the miR16 RT probe, that cluster disappears and there's no effect on the other clusters. So using this triplex assay, titrations were done in replicate to show in this series the counts for miR16, in the middle for miR21, and on the right the counts for GAPDH. And as seen before, these are highly reproducible and linear. So in summary, the RainDrop digital PCR system enables true single molecule counting with high sensitivity, assay multiplexing, with a closed tube design and a very simple workflow. This works for many different sample types. I've shown you some data using DNA. I haven't shown you data, but this works very straightforwardly if you pipette either cDNA or bisulfite-treated DNA into the well for counting. And what I hope I've illustrated is this works extremely well for counting a variety of species of RNA in a direct, quantitative way. So with that, I'd like to thank you for your time and open the webinar up to questions from the field.
Thank you. Our first question is, what is the minimum and maximum amount of RNA and DNA to test it?
So that's a great question. It's very straightforward to put limits on the system when you're counting DNA, because the typical mammalian cell has 2 copies of any species of target. For instance, if you have BRAF, one cell will have 2 alleles. So you can count the number of genome equivalents and be very clear as to how much DNA corresponds to how many genome equivalents. So a single human cell is 6.6 picograms of DNA, which is 2 alleles. So 3.3 picograms should give you 1 count. As I described, we have the dynamic range to look at up to 1 million single molecules. So you can load up to 3.3 or even more micrograms of input DNA and be in the single molecule range. For RNA, it's quite different. A single cell has a variety of different expression levels for different targets. So as long as the total number of RNA species that you want to interrogate in duplex, in singleplex, or in multiplex is in that range of about 1 million molecules, you're going to be able to do these single molecule counting experiments. And what I showed in the table was a range that went from hundreds of nanograms down to picograms.
Thank you. Another question is, what is the reaction volume you load onto the chip itself?
Thank you. Yes. So you can load a variety of volumes, anywhere from 25 microliters up to 50 microliters per well. As I described, every microliter that's pipetted into the well is pushed into the microfluidic device and turned into 5 picoliter sized droplets. The droplet size is precisely measured and is maintained from the beginning of the run to the end of the run using feedback control. So you can generate anywhere from 5 million droplets to 10 million droplets. Next question.
Next question. Has the system been used to quantify genomic DNA from plasmid preps or to quantitate plasmids directly from preps?
That's a great question. So certainly any DNA that you pipette into the well can be quantified. So we certainly have at RainDance used plasmids as DNA input, and certainly customers have done that as well. There really are not any limitations on the type of sample, DNA, cDNA, methylated DNA that has been bisulfite treated, and now I've shown you a variety of different RNAs. Whatever you pipette into the well, as long as there's a handful of molecules, we'll be able to count them.
Is it possible to use other real-time technologies other than TACMan?
Yes, I tried to point that out in one of the slides, that the system is open to a variety of reagents, and that's true both for the master mixes as well as the detection modalities. So certainly TACMan, we've shown worked a variety of different TACMan, but many other hydrolysis probe-based systems from a variety of vendors, and also intercalating dyes also work very well in the system. And in fact, it's the openness of the platform. All you're doing is pipetting 50 microliters or 25 microliters of sample plus assay into that well. The system will make droplets from it. And I did not show you methylation quantification. We've run kits from a variety of vendors that do all kinds of different things, including restriction digestion methods. And here we've shown a variety of one-step RT-PCR systems. The system is open, and in fact, that's one of the real advantages, is a researcher can experiment and play around with the system very, very easily.
What are the 4 one-step RT kits you mentioned you tested?
That's a great question. There really was not any prioritization or selection of that in any way. What I used were a couple of kits from Life Technologies. The one that I showed most of the data on was the standard superscript 3 platinum tack. But I also used kits from QIAGEN and from Quanta, and a second kit from Life Technologies as well, which is called AgPath. There was no kit that I tried that did not work. I just tried 4. Next question, please.
To determine the number of miRNAs in body fluids, how would you normalize?
You know, that's a really interesting biological question, and it actually is a very challenging one. The reason why I showed a couple of different controls for the miRNA experiments, both endogenous controls and exogenous spiking controls, was really to address that exact question. I think that this is something for panels of biomarker, candidate biomarker microRNA. You're really looking for changes in the ratios between those microRNAs, but there is a lot of power that can be added, both to use potentially message RNA as an internal control, but certainly the spike-in exogenous material really is useful at a number of levels, including making sure that your RNA purification was efficient. Any material that you spiked into the workflow at the beginning of the process, we can count exactly in the system, and any deviation from what was put into the process can then be used for normalization.
What kit was used for the microRNA data?
As I described, I didn't want to say this is the only system that can be used, but commercially for measuring microRNA using qPCR, the TACMan assay from Life Technologies was used for this. And I hope I was able to illustrate the reason why this is a very interesting product is that the microRNA, because it is short, doesn't give you a lot of room for both primers and probe to hybridize if you only have 22 base pairs. And that was one of the key features for this particular product that, as I mentioned, has been out on the market for almost 10 years and is well established, but has not so far been shown to be able to work in a one-step method or in multiplex. And for the RainDrop system, this was used right out of the box. They just work.
Can the assays be multiplexed to look at more than one virus or drug-resistant variants?
Yes. So when it comes to multiplexing, I showed you a number of multiplex assays, what I call triplex assays or fourplex assays. We demonstrated up to 10-plex using this single molecule formatting where you can use 2 colors and several intensities for each. It's extremely easy to put together 4-plex assays or even 5-plex assays. To do additional multiplexing beyond that will require a little bit of optimization, but it's very straightforward. A couple of runs, really all you're trying to do is add the 2 different probes for, let's say, target number 6 at the concentration so that at endpoint it's going to show up on that scatter plot in a distinct cluster. The software provided with the RainDrop system essentially takes those scatter plots, the raw data, and you can draw any gate around any spot on that scatter plot that you want, and the software will just immediately tell you how many molecules are present inside that gate. So as long as the clusters are separable enough to draw clean gates, you can continue to multiplex.
Is it true the Thunderbolt panels run on the RainDrop system?
Yes, and that's an interesting question. It was not the focus of this talk, and maybe there'll be a future webinar on that. The Thunderbolt cancer panel is a very unique product using just the source instrument, and it prepares samples that are as low as 10 nanograms of FFPE material for targeted multiplex sequencing on an Illumina platform. The data is we've been just launched this product. The data is very impressive in terms of the basic sequencing metrics, high depth with very consistently uniform number of reads per target. It's a very straightforward application, and I encourage people who are interested in using the digital PCR system for sequencing sample prep to go to our website. I believe it's info@raindancetech.com, and there'll be lots of material there to illustrate our Thunderbolts panel.
Okay, those are all the questions that we have time for today. Thank you for joining us. If you have any further unanswered questions, please send them to info@raindancetech.com, and we will get back to you as soon as we can. Thank you.